The ubiquitinated protein hears the beating of the drum. In the proteasomal cap, it knows its time has come. Stripped of its ubiquitin, stretched out like a string, pushed through the meat grinder, through the alpha 7 ring. In the dreaded beta chamber, it meets the long knives. 2 beta 1s, 2 beta 2s, and yes, 2 beta 5s. The proteasome gives, but the proteasome takes. It cleans up after the ribosome's mistakes. The protein is dead. Lower it to the ground. Long live the proteasome, let the trumpet sound. The creator, the destroyer, they are but one and the same. Please bow to the proteasome, lord of the peptide domain. A virus slithers in like a venomous snake. Untold death and destruction lie in its wake. Through the Sex61 channel, the class I molecule arrives. Into the ER lumen, the tap transporter drives some peptides, 8 residues in length preferred. Peptides generated as the ribosome erred. Tapase links tap to empty class 1. It can go to the surface. Now its peptide has come. A CTL spies its peptide in a class 1 groove. Therein lies a cell it must readily remove. Death to the cell, so that its sisters may thrive. Death to the cell, so that the host may survive. The cell is dead. Lower it to the ground. Long live the CTL, let the trumpet sound. The creator, the destroyer, they are but one and the same. Please bow to the proteasome, lord of the peptide domain. Every nucleated cell in the body has on its surface a billboard protein called an MHC class I molecule. So MHC class I molecules are on the surface of every cell, and these proteins exist in order to present peptides to the immune system. And the reason why we need every cell, every nucleated cell, so cells other than red blood cells basically, to actually express these proteins is because every cell in the body could be infected by a virus, and every cell could potentially become a tumor. And we need to be able to present viral peptides on the surface of the cell, or peptides from a tumor perhaps on the surface of the cell, so that the immune system, so cells called cytotoxic T lymphocytes, CTLs, can now recognize this peptide on the surface of this billboard protein, identify the infected cell perhaps, and then eliminate the cell. So that's the reason why we have MHC class I molecules. Now, the process by which MHC class I molecules acquire these peptides and take them to the cell surface is called antigen processing and antigen presentation. And processing refers to the generation of these peptides from the protein that is marked for degradation. So now, if you just think of any protein, so all proteins, like living creatures, though these are not creatures obviously, every protein has to be born, every protein has to die, and every protein breathes when it's around. So the protein is, if a protein misfolded, let's say by chance, as it was made in the ribosome, or if a protein is marked to be degraded because of a signaling pathway marking it, that protein is going to be tagged with a small 76 amino acid protein called ubiquitin. And ubiquitin, so we have to attach about 4 or more ubiquitins to a protein in a particular way to mark them for degradation. So when the protein is tagged for degradation, it can now be recognized by a structure called a proteasome. And the proteasome is made up of 4 rings, and the outer rings are called alpha rings, the inner rings are called beta rings, and the beta rings contain 3 proteases, so 3 proteins called beta 2, beta 1 and beta 5. And now each of these rings of the proteasome contains 7 proteins. So the proteasome also has, outside the rings, it has a cap at either end. So the ubiquitinated protein, this ubiquitinated protein is recognized by a protein in the cap. The ubiquitins are taken off. The protein is then using energy stretched out like a string and then pushed through the meat grinder that the proteasome represents through a pore. And as the protein emerges, the ubiquitinated protein that was de-ubiquitinated, unfolded and then pushed through the proteasome, it emerges as peptides. And these peptides vary in length, and the length we need for class 1 molecules are 8 or 9 amino acids long. So we have these peptides that emerge from the proteasome, and then they are transported into the endoplasmic reticulum because proteins are being synthesized. So proteins destined for the cell surface, for example, are being synthesized in the endoplasmic reticulum. So in the lumen of the endoplasmic reticulum, we may have a partly folded MHC class 1 molecule. It has the heavy chain, a light chain, and it doesn't yet have its peptide. And this partly folded class 1 molecule is linked to the trap transporter through which the peptides are coming by means of a few proteins, including terpacin, which make up the peptide loading complex. And the job of the peptide loading complex is to identify when a class 1 molecule has received a tightly binding peptide. So only when a peptide binds tightly to MHC class 1 will it be released from this peptide loading complex, and then this MHC class 1 molecule with the bound peptide will bud out in a vesicle from the endoplasmic reticulum. This vesicle will then go and fuse with the Golgi stacks, and as it passes through the Golgi through budding and fusion events, it will emerge from the Golgi, and then the vesicle that buds out of the Golgi will make its way to the cell surface, fuse with the membrane, the plasma membrane, and now the MHC class 1 molecule will be displayed on the surface with a peptide in its groove. So this overall is the process of antigen processing and antigen presentation.